Bismillah Rahman Rahim. We start the second video on November 2024, question paper 4.1. Question number 5. Lunularia cruciata is a primitive plant. Its body consists of a flattened sheet of photosynthetic tissue called a thallus. Figure 5.1 shows L cruciata with two different types of reproductive structures labeled A and B on its surface. Look at the diagram carefully. Figure 5.1. Structures A and B and the thallus of L cruciata are haploid. Explain what is meant by haploid. Very simple. Haploid means one set of chromosomes, or you can say have n number of chromosomes, or you can say half the diploid number, or you can say chromosomes not in homologous pairs. Each chromosome is different in size, shape, gene, loci. So basically very simple answer, one set of chromosomes. I prefer the answer of only one of each pair. Have n number of chromosomes. N is the terminology that we use for haploid, and for diploid we say 2n. So we either say n diploid or 2n. I'm just trying to explain it to you. And each chromosome is different in size. Then coming to the B part of the question, structure A contains pale discs of tissue C that can germinate into new L cruciata. The new plants that develop from C are genetically identical to the parent plant in figure 5.1. Structure B contains male sperm that are chemically attracted to swim to female eggs on a neighboring parent plant. When the egg and sperm fuse, they form structure D. Structure D develops to produce spores that grow into new plants that are genetically different from the two parent plants. Identify which of the structures A, B, C, and D are associated with sexual reproduction, B and D, right? And produced by mitosis, A, B, C, D. And the site of meiosis is only D. So each letter may be used once, more than once, or not at all. So everybody has to be clear, you see, associated with sexual reproduction, B and D. Why B and D? B contains male sperms that are attracted to female on a, when the egg and sperm fuse, they form structure D. So B and D are required in sexual reproduction, because what is sexual reproduction? It is the fusion of the gametes to form the zygote. And produced by mitosis A, B, C, and D. Because the newts that develop from C are genetically identical to the parent plant. The site of meiosis, why are we saying D? Structure D develops to produce spores that grow into new plants, and it said here when the sperm and egg fuse, they form structure D. And structure D develops to produce spores that grow into new plants that are genetically different from the two parent plants. So a little tough for the question. I agree with you if you say it was tough. Then C, figure 5.2 shows a horse, Equus caballus. Horses are diploid animals that reproduce sexually. Male and female horses produce gametes which fuse to form genetically different offsprings. Explain the need for reduction division during meiosis in the production of gametes in animals such as horses. I mean, of course, this is basically to maintain the chromosome number, the diploid number. So from parents to offspring, from generation to generation, to give us genetic variation in the gametes. And because too many extra sets of chromosome can cause problems. So you cannot afford to have extra sets of chromosomes. So meiosis results in haploid gametes being formed. And when they fuse and the zygote is formed, then the diploid numbers is restored. Otherwise, in every generation, the number would have doubled. You see, if two diploid cells fuse, then this would result in a 4n cell. 2n plus 2n will be 4n. And the zygote will have 4n. The next generation it will be 8n, then 16n. So the chromosome number will keep on increasing in every generation. And that, of course, will result in massive problems. If you just have one extra chromosome, it results in Down syndrome. And it's not normal. The person is not normal, the person who has Down syndrome. So basically, you've got to maintain the chromosome number from generation to generation. So it results in genetic variation because the gametes are different and extra sets of chromosome can cause problems. So we're preventing that. Then coming to question number 6, the mammalian kidney is responsible for the excretion of urea, osmoregulation, the homeostatic control of the water potential of the blood. Outline how and where the excretory product urea is made in the body. 2 marks. Excess amino acids are deaminated. Where? In the liver. So the fact that you wrote excess amino acids are deaminated got you 1 mark. And if you didn't know the word deamination, you could have said the excess amino acids have the amino group removed. If you didn't remember the word deamination, you could have written that amino group removed. Where? In the liver. Then part 2, homeostatic control of the water potential of blood includes receptors, effectors, and target cells. Identify the names and locations of these components of homeostatic control in osmoregulation. And that was 3 marks. Osmoreceptors are in the hypothalamus. Effectors are in the collecting duct, or you could have said the distal convoluted tubule. Even nephron was acceptable by Cambridge. And target cells are the collecting ducts. So osmoreceptors in the hypothalamus, effectors are the collecting duct, and the target cells are the collecting duct cells where all the water, you see, the water will be reabsorbed. So less water, the urine will become concentrated because it will contain less water. Of course, the urea will be the same amount, but because it's in less water, so that's why we say the urine becomes concentrated. So wording it is the main thing. You might know the topic, but you don't know how to word your answer. Well, there you are then. So excess amino acids deaminated where? In the liver. Osmoreceptors in the hypothalamus, effectors in the collecting duct, and target cells are the collecting duct cells, where of course the aquaporins go and get attached to the side of the lumen, and the water molecules are then absorbed and pass into the blood to then maintain the concentration of the blood plasma. The glomerulus and Bowman's capsule of the nephron are important in the formation of urine. Outline the role of the glomerulus and Bowman's capsule in the formation of urine. Again, a very direct question. You must know this, and that was very easy. Ultrafiltration results in the glomerulus and forms the glomerular filtrate. High hydrostatic or blood pressure in the glomerulus. So water and the solutes move out of the glomerulus into the Bowman's capsule. And why does that happen? Because of two things. Number one, the basement membrane is there, but it's okay. But there are fenestrations or holes in the endothelium, and they are more than the normal. And then the podocytes are holding that membrane up. So all this detail would have got you your 3 marks. So all this is how you had to write it, and I'm sure you all can easily remember it, and you should also memorize some of these points which are direct questions so that you can do any question on this chapter. Now coming to the C part of the question, concentrated urine contains a high concentration of solutes and a small volume of water. Different species of mammals vary in their ability to produce urine with a high solute concentration. Table 6.1 compares the ratio of the solute concentration of urine U to the soluble concentration to the solute concentration of blood plasma P in some mammal species. The habitats of the mammal species are also shown. So beaver has a maximum ratio of solute concentration to solute of blood plasma is 1.7:1. And in the habitat is rivers and lakes. Then humans 4.5:1 variable, camel 8:1 desert, rat 9:1 variable, kangaroo rat 16:1 desert. With reference to table 6.1, suggest what the different values of UP show about the ability of these mammal species to tolerate a shortage of water in their environment. And that's 3 marks. So basically very simple. Higher UP ratio can tolerate water shortage. Higher UP ratio 18:1 kangaroo rat desert. So higher UP ratio can tolerate water shortage. Higher UP ratio means what? Much water reabsorbed, so small volume of urine produced. And the highest UP ratio can concentrate urine, so it is very well adapted to dry environment. So you have to come up with 3 marks and 3 points, and we've got 3 points which you could have come up with. When in the question it says with reference to table 6.1, then please do refer to this. Read the table again and then write your answer in continuation of the table. So higher UP can tolerate water shortage, higher UP much water reabsorbed, higher UP can concentrate urine. This all these things we got from the table, nothing of some information that you knew before. It's just also information from the table. Now coming to question number 7, Spia multiplicata is one of the several species of American spadefoot toad. Young spadefoot toads are called tadpoles and live in water in ponds. S. multiplicator tadpoles show three different phenotypes due to genetic variation. The three phenotypes are detritus feeder, intermediate, and carnivore. Detritus feeders are small, carnivores are large, intermediates vary in size between the two extremes. A detritus feeder and a carnivore are shown in figure 7.1. So we have a detritus feeder and we have a carnivore. Now, detritus feeders eat detritus, small pieces of dead organic matter and algae, have smooth mouthparts, small jaw muscles, and long intestines. Intermediates can eat all available food, detritus, algae, and fairy shrimps, have teeth-like mouthparts. These have smooth mouthparts, medium-sized jaw, small jaw, and medium-sized intestine. These have long intestines. Then carnivores eat fairy shrimps and other small animals. They eat all the available food. They eat only small pieces of dead organic matter and algae. Have teeth-like mouthparts. These also have teeth-like mouthparts. Large jaw muscle. They had medium jaw muscle and short intestine. So we have long intestine, medium intestine, and short intestine. Scientists counted the number of each type of tadpole in two different ponds, pond 1 and pond 2. In pond 1, the scientists observed a high density of tadpoles, a low abundance of food, that most of the tadpoles they counted were either detritus feeders or carnivore with very few intermediates present. Describe and suggest explanation for the type of natural selection that appears to be acting in pond 1. Now, what sort of a selection is this now? What you have? You have disruptive selection. Why is it disruptive selection? Because extremes, carnivore and detritus feeders survive, and there is high competition or intense competition. Intermediates are outcompeted for, don't get enough of the detritus or algae and fairy shrimp or small animals. So lack of food is, or the limited food is selection pressure. So we have two different categories which are at an advantage and one category which is at a disadvantage. So high density of tadpoles, low abundance of food, and most of the tadpoles were either detritus or carnivore and very few intermediates. So we have the three varieties. Two were successful and one was not so very successful. That is why in disruptive selection you get a graph like this. So there are two peaks and then there's a dip here, and that is disruptive selection. So wording it: disruptive selection, extreme survive, intermediates die, high competition, intermediates don't get enough detritus, lack of food is the selection pressure. In pond 2, the scientists observed a low density of tadpoles, sufficient food availability for all tadpoles, that most of the tadpoles they counted were intermediates with fewer detritus feeders or carnivores. Describe and suggest explanation for the type of natural selection that appears to be acting in pond 2. In pond 2, it's stabilizing selection. Why is it stabilizing selection? Because you see the extremes, carnivore and the detritus feeders die and are selecting against. And intermediate survive. Only the one survive, the intermediate survive. So there is less competition. Intermediates can eat both types or a wider range of food. Greater variety in the diet improves the growth and development of the intermediate tadpoles. So most of them were intermediates. So the intermediates are the success story. Now this is called stabilizing selection, because the one type has now gained is sort of gained more popularity, and it is an advantage, and that's why they are growing more, and that's why there are a larger number of them now in this pond. So wording it now: stabilizing selection, even if you didn't know that word, but you've said extremes are selected against and intermediate survive, less competition, intermediate can eat a wider range of food, greater variety in the diet improves growth and development of intermediate tadpoles. Then coming to the part 3 of the question, the intestine length of L's multiplicator tadpole shows continuous variation. Sketch a curve to show how intestine length varies in the tadpole population in pond 2. Now we have the length of intestine and we have the number of tadpoles, so it's going to be something like this. Because the length of the intestine is the number of tadpoles and the length of the intestine is a normal distribution curve. So there's 1 mark for that, and you got your 1 mark if you drew this for me. A student suggested that the variation in S multiplicator tadpoles could lead to sympatric speciation in some populations. Outline the features of sympatric speciation. Now, please remember this has changed in the new syllabus, and if you gave me the old definition, I won't give you any marks. Now, in this, you say new species formed due to reproductive isolation caused by ecological barrier and in the same geographical region. So there is a behavioral or an ecological barrier, and it has to be in the same geographical region. So sympatric speciation is always in the same place, and the reason why there is going to be sympatric is because there's going to be some behavior, or they don't mate, or there's going to be some other ecological separation. They may be in two different parts of the pond, and they do not intermingle and do not breed. So new species formed due to reproductive isolation caused by ecological or behavioral separation. You could have also said isolation, and it has to be in the same geographical region. You see, allopatric speciation, which is the opposite of this, has to be it has to be a geographical isolation. It has to be a geographical barrier between the two, and that results in allopatric speciation, which is not in this question, but it could come in another question this year. Then coming to the B part of the question, figure 7.3 shows the evolutionary relationships between three species of American spadefoot toad, Spia multiplicata, Spia hammondi, and Spia bombifrons. And time in millions of years ago, 60, 40, 20, 0. Now explain how analysis of DNA allowed the evolutionary relationship shown in figure 7.3 to be determined. And that's 3 marks. Well, basically what you've got to understand is how can DNA help us in that. So it's a very simple, this come in so many papers. DNA sequences, we try to look at the DNA sequence of all the 3 species. And then we count the nucleotide or the base differences and the similarities. And the fewer the difference means they're more closely related. The fewer the differences, I mean, they're only difference of say 10 bases, and then another there's a difference of 40 bases. So the fewer the differences means they're more closely related, less time since divergence or less time to common ancestor. And then we have the bioinformatics, which is a database or a software which is also called BLAST, BLAST, which we can help us to then compare the data. And the S. hammondi and the S. bombifrons have the fewest genetic difference, are the most genetically similar. So we study the DNA sequence, we find the nucleotide differences. Fewer the differences means they're more closely related. Bioinformatics helps in further analyzing the whole data. And S. hammondi and S. bombifrons most genetically similar. Now these are these two. Coming on to question number 8, scientists use many different techniques in genetic engineering. Sometimes the gene for genetic engineering cannot be extracted from the donor organism. Instead, the gene is synthesized using one of the 2 different methods. Outline the 2 methods of synthesizing a gene for use in genetic engineering. Make the cDNA from messenger RNA using reverse transcriptase, or you synthesize the gene chemically from nucleotides or join the nucleotides using known amino acid sequences from the database. So make cDNA from mRNA using reverse transcriptase, synthesize gene from nucleotides using amino acid sequence from database. So if you know the amino acid sequence, then you can know the DNA sequence. So that is, we've got all the database for that. Then we come on to B part of the question. DNA ligase and DNA polymerase are 2 enzymes that are used in genetic engineering. Complete table 8.1. To show the roles of DNA ligase and DNA polymerase in genetic engineering, use a tick if the enzyme has a role or a cross if the enzyme does not. So joins 2 sections of sugar phosphate backbone in DNA, we use DNA ligase. 2 sections of sugar phosphate backbone in DNA. So we use the DNA ligase, not the DNA polymerase. Then adds a gene to the plasmid. Then we use DNA ligase. We do not use a DNA polymerase. Adds free activated DNA nucleotides to a polynucleotide. That is, of course, the DNA polymerase. So DNA ligase is only used to make the sugar phosphate backbone. to join it when we're using two different, when we're joining the plasmid with the plasmid is being used as a vector. Part C of the question is a direct question. The PCR polymerase chain reaction is used to make many copies of a gene. Three temperatures used in a PCR cycle. The state the three temperatures that are used and outline what happens at each temperature during a PCR cycle. Now, it's very simple. 90 to 98 degrees Celsius, DNA denatures or the strands separate. 50 to 65 primers bind to the base pairs, or you say anneal to the DNA. And 68 to 75, then you use the Taq polymerase or the DNA polymerase, which makes the DNA, the new strand of DNA. So exactly 3 marks, and then you give me all these points. 90 to 98 DNA strands separate, then 50 to 65 use the primers bind, or we call it annealing, anneal to the DNA. And then 68 to 75 is the Taq or the DNA polymerase, which makes the new DNA strand. So that is why polymerase chain reaction results in you can make many copies of the gene or that little bit of DNA that you want more copies of it, so you want to study it.